I'm Maryann Robin, and I'm going to be talking to you about the role of the liver and pancreas in the digestive system. Today we are going to be talking about the role of the pancreas and liver in regulating digestion. We are going to start with the pancreas, but first I wanted to say a quote out of Silverthorne that emphasizes the importance of the pancreas and liver in the digestion process. In chapter 21.6, it says, each day the liver, pancreas, and intestines produce more than 3 liters of secretion whose contents are necessary for completing the digestion of ingested nutrients. This can help us infer that the liver, pancreas, and intestines have a great role in digestion. Now let's start discussing the pancreas. The pancreas is an exocrine and endocrine organ. The exocrine and endocrine secretions help aid in digestion. The exocrine portion of the pancreas makes a pancreatic juice of enzymes to break down sugars, fats, and starches. It contains digestive enzymes and a water solution of sodium bicarbonate. The endocrine portion helps the digestive system by making hormones to regulate your blood sugar levels, appetite, stimulate stomach acids, and it also tells your stomach when to empty. Digestive enzymes are produced by the exocrine pancreas. Most bicarbonate, which is a digestive enzyme, comes from the pancreas and is released in response to neural stimuli and secretin. The exocrine portion of the pancreas consists of lobules called acini, similar to salivary glands. The ducts from the acini empty into the duodenum, which is the first part of the small intestine after the stomach. Acinar cells secrete digestive enzymes, and the duct cells secrete the sodium bicarbonate solution. The exocrine portion of the pancreas is also known to help digest proteins such as trypsin and chymotrypsin, carbohydrates like amylase, and lipase, which is considered as a fat. Islets are clusters where the endocrine secretion comes from. They include the hormones insulin and glucagon, which enter the bloodstream. Most pancreatic enzymes are secreted as zymogens that must be activated once they arrive in the intestines. The activation process begins when the brush border enteropeptidase, which is an intestinal enzyme that activates trypsin, converts inactive trypsinogen to active trypsin, allowing trypsin to convert other pancreatic zymogens to their active forms. In order for the pancreas to release enzymes, it needs signals. The signals are distension of the small intestine, the presence of food in the intestine, neural signs, and the gastrointestinal hormone CCK. The enzymes then enter the intestine in a watery fluid that also contains bicarbonate to help regulate digestion. Now let's talk about the importance of bicarbonate secretion. It is important because when it is secreted into the duodenum, it neutralizes the acid entering from the stomach. The production of bicarbonate requires the enzyme carbonic anhydrase in levels similar to those found in the renal tubule and red blood cells. Chloride is also important for bicarbonate secretion. It enters the cells by indirect active transport and leaves from the apical side through a CFTR channel. If there is a defect in the CFTR channel, this can lead to cystic fibrosis. One of the hallmark signs of cystic fibrosis is the disruption of pancreatic secretion, which is why it is so important to understand. Sodium and water secretion are a passive process in the pancreas, which are driven by electrochemical and osmotic gradients. Sodium is able to move down the electrochemical gradient through leaky junctions between the cells. Sodium and bicarbonate form the extracellular fluid transfer into the lumen by an osmotic gradient, and then water follows, which leads to the watery solution of sodium bicarbonate. Now let's discuss the liver. The liver is the largest internal organ, weighing about 3 pounds. One of the most important things about the liver is that it produces bile, which is released from the gallbladder as a non-enzymatic solution that facilitates the digestion of fats. There are 3 key components of bile: bile salts, bile pigments, and cholesterol. Bile salts facilitate enzymatic fat digestion, and are made from steroid bile acids combined with amino acids. Bile pigments like bilirubin are waste products of hemoglobin digestion, and cholesterol is excreted in feces. All pharmaceutical drugs can be cleared from the body by hepatic processing and excreted in bile. Bile that is secreted from hepatocytes, which are liver cells, go down the hepatic ducts to the gallbladder, which stores and concentrates bile solution. When bile salts reach the terminal section of the ileum in the small intestine, they encounter cells that reabsorb them and send them back into the circulation. When bile salts make it all the way to the colon, they're converted back to bile acids by bacteria in the colon and then recycled back to the liver by the hepatic portal vein. This recycling of bile salts is important for fat digestion because the body's pool of bile salts have to cycle 2 to 5 times for each meal. Usually bilirubin and other wastes secreted in bile can't reabsorb and are excreted in feces. At the bottom of the slide, we can see how hepatocytes are organized in the liver into irregular hexagonal units called lobules. Here we can find a flowchart explaining the importance of the liver. The liver has many functions such as glucose and fat metabolism, protein and hormone synthesis, urea production, detoxification, and storage. We can see that the hepatic portal vein and hepatic artery transport materials to the liver from the gastrointestinal tract or from metabolites and drugs from peripheral tissues. We can also see that the bile duct and hepatic vein transport materials away from the liver, either to the duodenum or the peripheral tissues. And now let's see how the digestive system works as a whole with this video.
Across the whole planet, humans eat on average between 1 and 2.7 kilograms of food a day. That's over 365 kilograms a year per person, and more than 28,800 kilograms over the course of a lifetime. And every last scrap makes its way through the digestive system. Comprised of 10 organs, covering 9 meters, and containing over 20 specialized cell types, this is one of the most diverse and complicated systems in the human body. Its parts continuously work in unison to fulfill a singular task, transforming the raw materials of your food into the nutrients and energy that keep you alive. Spanning the entire length of your torso, the digestive system has 4 main components. First, there's the gastrointestinal tract, a twisting channel that transports your food and has an internal surface area of between 30 and 40 square meters, enough to cover half a badminton court. Second, there's the pancreas, gallbladder, and liver, a trio of organs that break down food using an array of special juices. Third, the body's enzymes, hormones, nerves, and blood all work together to break down food, modulate the digestive process, and deliver its final products. Finally, there's the mesentery, a large stretch of tissue that supports and positions all your digestive organs in the abdomen, enabling them to do their jobs. The digestive process begins before food even hits your tongue. Anticipating a tasty morsel, glands in your mouth start to pump out saliva. We produce about 1.5 liters of this liquid each day. Once inside your mouth, chewing combines with the sloshing saliva to turn food into a moist lump called the bolus. Enzymes present in the saliva break down any starch. Then your food finds itself at the rim of a 25 centimeter long tube called the esophagus, down which it must plunge to reach the stomach. Nerves in the surrounding esophageal tissue sense the bolus's presence and trigger peristalsis, a series of defined muscular contractions. That propels the food into the stomach, where it's left at the mercy of the muscular stomach walls, which pound the bolus, breaking it into chunks. Hormones secreted by cells in the lining trigger the release of acids and enzyme-rich juices from the stomach wall that start to dissolve the food and break down its proteins. These hormones also alert the pancreas, liver, and gallbladder to produce digestive juices and transfer bile, a yellowish-green liquid that digests fat, in preparation for the next stage. After 3 hours inside the stomach, the once shapely bolus is now a frothy liquid called chyme, and it's ready to move into the small intestine. The liver sends bile to the gallbladder, which secretes it into the first portion of the small intestine, called the duodenum. Here, it dissolves the fats floating in the slurry of chyme so they can be easily digested by the pancreatic and intestinal juices that have leached onto the scene. These enzyme-rich juices break the fat molecules down into fatty acids and glycerol for easier absorption into the body. The enzymes also carry out the final deconstruction of proteins into amino acids and carbohydrates into glucose. This happens in the small intestine's lower regions, the jejunum and ileum, which are coated in millions of tiny projections called villi. These create a huge surface area to maximize molecule absorption and transference into the bloodstream. The blood takes them on the final leg of their journey to feed the body's organs and tissues. But it's not over quite yet. Leftover fiber, water, and dead cells sloughed off during digestion make it into the large intestine, also known as the colon. The body drains out most of the remaining fluid through the intestinal wall. What's left is a soft mass called stool. The colon squeezes this byproduct into a pouch called the rectum, where nerves sense it expanding and tell the body when it's time to expel the waste. The byproducts of digestion exit through the anus, and the food's long journey, typically lasting between 30 and 40 hours, is finally complete. TED-Ed is a nonprofit. If you value our work, please consider supporting it on Patreon.
And that is the conclusion of my presentation of the digestive system with the liver and pancreas. Thank you.